Welcome viewers. Today I will discuss on the recombinant DNA vaccine. I will tell you how vaccines work, that is the mechanism of vaccination, different types of recombinant DNA vaccines, and advantages and disadvantages of recombinant DNA vaccines. Vaccine, as we know, gives us protection from an infectious agent or disease, and it contains either whole organism or part of the organism which activates the immune system. Now, how vaccines work inside our system? When the agent present in a vaccine enters into our system, it is engulfed by the phagocytic cells present in our system. Mainly macrophages, dendritic cells, they engulf the agent present in the vaccine, and then they process and present some part of it on their surface. T-lymphocytes, both T helper or TC or cytotoxic T lymphocyte, they are activated by these foreign pathogens or by these fragments which are foreign to them, and they are activated. These activated T lymphocytes then differentiate into effector T lymphocytes and to memory T lymphocytes. On the other hand, simultaneously, B lymphocytes also are activated by the agent, and B lymphocytes differentiate into effector B lymphocytes and memory B lymphocytes. These memory T cells and B cells, they keep the memory of that particular agent, and when in future the same agent enters into the system, they protect the system from that invading agent. This is the mechanism of immunization. Now, immunization can be achieved naturally through natural infection or artificially through vaccination. So, vaccination is an example of artificially acquired active immunization. Immunization was developed by English physician Edward Jenner in 1796. During late 70s, Edward Jenner noticed that milkmaids who had suffered from cowpox, they never catch the smallpox virus. So, what he did, he took the cowpox lesions and inoculated it in a healthy boy. And purposefully, after 6 weeks, he inoculated the boy with smallpox virus. And as predicted, the boy did not suffer from smallpox. Edward Jenner named these techniques vaccination, and very quickly these techniques spread throughout Europe. Later, Louis Pasteur developed the idea of vaccination, and he developed many vaccines. Now, when scientists thought about designing vaccines, they first thought of whole organism, either live attenuated or killed, inactivated or killed microorganism. They found that use of live attenuated microorganisms as vaccinating agent has a number of advantages. Like, since these microorganisms grow inside the system, inside the host, immune system gets enough time to recognize them, and the immune response generated is more prominent and strong. Whereas the disadvantage of this type of vaccine is that they can revert to their virulent form, and these live attenuated organisms can cause the disease in immunocompromised patients. Then they thought of killed microorganisms as vaccinating agent. Advantage was these vaccines are safe, but the disadvantage is, since the organism is not growing inside the system, several doses are required to properly activate the immune system. So, scientists then thought of designing vaccines with purified macromolecules, and these vaccines are called second generation vaccines. The advantages are, these vaccines are safe compared to the first generation vaccines, and since immune system is developed against specific antigens, it lowers the adverse effects of that molecule. And disadvantage is immune system activation is a little difficult here, and they do not activate the T helper cells. Hence, scientists pay attention on developing some vaccines which are safe, at the same time more efficient than the first and second generation vaccines. So, they took the help of recombinant DNA technology and developed vaccines using this technology. These vaccines are called third generation vaccines or recombinant DNA vaccines. So far, 3 types of recombinant DNA vaccines have been developed, but all of them are in experimental phase. The first type is called recombinant vector vaccine or live genetically modified organisms. Second is genetic vaccine or DNA vaccine, and third type is recombinant inactivated vaccine. Recombinant vector vaccines are developed using an organism and deleting some of the disease causing genes from it and introducing one or more gene of interest from a pathogen. Now, this organism will act as a vector. When given into a host, it will replicate inside the host and it will produce the foreign proteins, and these foreign proteins will activate the immune system. So far, studies have shown that many viruses like vaccinia, canarypox virus, adeno-associated virus, many attenuated strains of salmonella, they can be used as these vectors. And vaccinia virus, that is the cowpox causing virus, it has a large genome, almost 200 gene encoding genome. So, if some of the genes are deleted and one or more gene of interest is inserted into it, the modified vaccinia virus will still replicate, can be used as a vaccinating agent. So far, vaccines against hepatitis B and herpes simplex virus has been developed using this recombinant vaccinia vector. Now, there are several advantages over the traditional vaccines. The recombinant vaccine is safer compared to the first and second generation vaccines. And the development of this vector is very simple and the cost is much cheaper. And also, more than one new gene can be introduced at a time inside a vector. Also, storage and transportation is very easy for this type of virus, and since these viruses, vector viruses, can be delivered through natural routes, they will develop both the systemic as well as mucosal immunity. The disadvantage of this vector vaccine is the use of vaccinia carries risk of adverse effects and the virus can easily spread to susceptible contacts. Now, second type of recombinant DNA vaccine is genetic vaccine or DNA vaccine. Here, directly the genetic material is used as the vaccine. The gene of interest, one or more gene of interest is inserted into an expression plasmid, and after construction of the expression plasmid, it is transformed into bacteria. Inside bacteria, the plasmid multiplies, and after that the plasmids are separated from the larger bacterial DNA and it is purified. This purified DNA is used as a vaccine. It is delivered through saline or through gene gun delivery into intramuscularly. In gene gun delivery method, the expression plasmids are coated with gold or tungsten particles. Then the gene gun introduces the DNA coated gold particles into muscle. Inside the muscle, these DNA molecules are taken up by the muscle cells. These DNA molecules are expressed to produce the proteins. Those proteins are recognized as foreign and they are presented on the surface of the muscle cells. Now the immune system cells recognize them as foreign and they mount an immune response. The advantage of this type of vaccine is, since the antigen is expressed in vivo, it will resemble the native epitopes. That means it will resemble the actual protein which is present on the organism. And plasmid vector construction is very simple, and it can be produced very quickly, and at a cost is much lower in this case. And DNA vaccine can encode more than one protein at a time. So, with a single dose, many genes can be introduced into the host. And very little plasmid is required to boost up the immune system. For gene gun delivery, 0.2 to 20 microgram plasmid is enough to activate the immune system. These vaccines are temperature stable and very easily they can be stored and transported. Now, the biggest disadvantage of DNA vaccine is that only protein antigens can be encoded in the DNA molecule. Many organisms have outer capsule made of polysaccharides, and polysaccharides cannot be coded in a gene. So, DNA vaccine could not activate the immune system against microbes having polysaccharide capsules. The third type of recombinant DNA vaccine is called recombinant inactivated vaccines. These vaccines are composed of mainly subunits like polysaccharides, toxins or glycoproteins present in an organism. These polysaccharides, glycoproteins, they can be made in laboratory synthetically. Then they are conjugated with protein and they are given into the host as a vaccinating agent. One more example of recombinant inactivated vaccine is virus-like particles. When one or more genes encoding structural proteins are expressed at the same time, they assemble to form the virus-like particle. These particles do not carry any genetic material, thus they do not replicate inside the host, but they look like the virus. So, immune system gets activated and antibodies are generated against the coat proteins. So, when the actual virus enters into the system, the virus can be prevented by the antibodies generated. Currently, virus-like particles have been generated against hepatitis B virus and human papillomavirus. The advantage of virus-like particles is that they do not require any inactivation treatment like formalin, treatment with formalin and detergents, because they are not active virus. They just look like a virus. They are not actually virus. And they can be produced very rapidly and within weeks. And this property is useful when a new pandemic strain emerges. Very quickly in laboratory, virus-like particles can be developed once the structural protein has been identified. Disadvantage is it is a new technique, so extensive study should be done before using this particle as a vaccine. And if we look into the time span required for vaccine production, then we will see that recombinant DNA vaccine production needs much less time compared to the traditional vaccine production. If traditional vaccine production requires 28 weeks for the simplest vaccine, then for genetic vaccine, the time span required is 18 weeks only. Among the vaccines developed so far, first generation, second generation and third generation, from the safety point of view, the DNA vaccines or the third generation vaccines are the safest one, because the DNA vaccines are composed of a single DNA molecule. It has no property of infection. It will only produce the protein. That protein will activate the immune system. So, this is the safest vaccine discovered so far. On the other hand, the least safe vaccine is live attenuated vaccine, because live attenuated vaccines always have a chance of revert back to their virulent form. And if we look from the efficacy point of view, then live attenuated vaccine is the most efficient one, because it is growing inside the host system. It is acting like a real infection. So this is the most efficient vaccine, but at the same time it is the least safe vaccine. Whereas subunit vaccine, virus-like vaccine, DNA vaccine, though they are not so efficient, but since they are not carrying any infectivity, they are the safe vaccines. So we can say third generation vaccines are safer compared to the first and second generation vaccines. Today we discussed different types of recombinant DNA vaccines and their mode of action, the advantages and disadvantages of different recombinant DNA vaccines. However, these vaccines are not available in market yet. They are undergoing some clinical trials, and hopefully within few years these vaccines will be available in market with more efficacy, more safety over the traditional vaccines which are available nowadays. Thank you.